I'm now pleased to introduce Drs. Odom and Barrett. Dr. Odom is from the University of Washington School of Medicine. He also received a grant in 2022 for the research into phenotypic screening of deimmunized DMD gene therapy vectors. His colleague, Dr. Philip Barrett, is a postdoctoral researcher at the University of Ottawa who is pioneering innovative approaches to evaluate DMD therapeutics using human stem cell-derived engineered muscle tissues. Their research is bridging the gap between preclinical testing and clinical translation, offering new insights into early disease mechanisms and providing more predictive human models for therapeutic development. Drs. Odom and Barrett are not able to attend tonight, so they have prepared a brief video update for us.
Good evening. My name is Guy Odom. I'm a principal investigator at the University of Washington in the Department of Neurology. I'm here to present an update on our research. This is a highly collaborative, multidisciplinary effort between the Odom and MAC labs. And there are 4 primary research components to this project. The first being vector and regulatory cassette validation. We have generated approximately 30 vectors that are essential tools in moving this research forward. We identified the most efficacious retroviral vector and the most active regulatory cassette out of those tested. Second, a primary objective of this project is to re-engineer microutrophin. As we published in 2020, that there are deficits in first-generation microutrophin function, so we address that by re-engineering utrophin and have tested 4 novel utrophins with 1 leading candidate that contains 6 spectrin-like repeats. This has been followed up with protein expression and metabolomics across 6 treatment groups, and we have a manuscript currently in preparation. Third, deimmunization of dystrophin. As a primary interest in blunting immune responses against dystrophin, we have deimmunized the spectrin repeats 16 and 17 of dystrophin to endow our new microutrophin clone with the capacity to localize nNOS to the membrane, which we feel is critical for therapeutic function. These clones have cover 4 MHC1 and 8 MHC2 allele types, so it's a broad representation in terms of represented alleles in humans. And each of these have been vectorized within the 6 repeat microutrophin. And the 4th component, I'll pass it over to Dr. Barrett, and he'll discuss human DMD iPSC-derived 3D muscle tissues.
Excellent. Hello. As Guy just said, I am a postdoctoral researcher in the MAC lab, and thank you for the opportunity to speak today. What we've done in our lab is develop a unique system for using human muscle tissue that we grow in the lab from stem cells. Think of it as having a tiny piece of human muscle, some healthy and some addicted. This lets us assess how well our treatments might work in humans before trying them in our models of patients. This is important as it allows us to focus our valuable time and resources on the most promising candidates and really assess a high volume of them compared to when you use these mammalian models. I'm happy to share that our initial results are pretty promising. The FDA-approved microdys5 construct is showing early signs of restoring function in lab-grown muscle, which is kind of a big deal because it validates the system. It shows that it causes no disastrous effects, and it acts as a positive control and a benchmark for testing these novel therapeutics that Guy has generated. And we are now at the point we are delivering these successfully into these human muscle, and we are starting the experiments to assess their functional recovery. So exciting times ahead. In addition, we're working with a private company called Curie Bio, and they're helping us to refine and scale up the system. This lets us evaluate, again, more and more of these therapies quickly and efficiently, but also gives us a pipeline to commercial application, because there's no point doing the work if we can't see the future of how to get to the product. This work is being achieved through the help of this grant. It's also benefiting many of the other projects around UW, including investigating some of these early moments in DMD progression and identifying new and improved therapeutic targets for the future. So it's going to have downstream effects that aren't just to do with this direct novel therapy. So to conclude, these grants are really helping us develop treatments more efficient and safer for a greater number of boys with DMD. So thank you for listening, and if you have any questions regarding this, please reach out. Thank you.